Okay guys, good evening. We are now back for lesson 4 part B of the basic calculation in microbial enumeration. In this section of part B, we are going to look into the basic calculation of dilution for microbial enumeration. For every microbial calculation or microbial enumeration must undergo dilution process in order to get a valid microbial counting that we need. As we have said already in the previous lecture, that the valid count is between 30 microbial cell or colony to 300 microbial cell or colony. And for you to do this, you have to get a dilution process, a dilution protocol in order to achieve this. And the two basic dilution protocol we have is the direct dilution process and the serial dilution process. In this direct dilution process, we usually use this direct dilution process when the concentration is known. When you have a microbial system that you know the microbial concentration, you have the microbial population, you standardize, the solution is standardized, the culture is standardized, then you use direct dilution in order to reduce the microbial population to have a valid count or valid culture plate for you colony count. So when you use direct dilution for concentration, while serial dilution process is for non-standardized microbial solution. That is when you don't know the concentration, you don't know the microbial load or microbial population within the system. You use serial dilution in order to systematically reduce the population to have a valid microbial count. And we know we have already discussed serial dilution in our in the physical class, where we now say serial dilution is a sequential method of reducing the microbial population within a system in order to have a valid culturable or valid microbial culture count, colony counting, whether it's a cell or the colony counting. So it's just a systemic method of reducing the microbial population in order to have a valid culture plate. So that is basically what is serial dilution. Without wasting much time, we'll go into direct dilution. Direct dilution protocol is based on the equation of C1 V1 equals to C2 V2, which is a basic equation in concentration recomposition in chemistry. Your chemistry 101 102, I think that is where you dealt with this C1 V1, the way of determining concentration or reducing a concentration or increasing the concentration of a solution. So the C1 is initial concentration, V1 is initial volume, C2 is final concentration, V2 is final volume. Please make no mistake about this concentration or trying to misplace the position or the value or what it means by initial concentration is different from microbial population of a system. Concentration is the number of cells per a given meal or a given volume, while the total population is the total population of that system. We'll discuss that with basic equation, I think in example 4 we'll discuss that. Then let's go to exercise 3. So I will discuss this direct dilution. Then let's say if 2 liters of a watermelon juice was found to contain 30 cells, a known concentration now of microbes, what will be the microbial cell concentration of the juice if 1 liter of distilled water was added to the juice? You have to be able to interpret the question very well in order to extract your values properly for you to have a simple calculation. But if you don't extract very well, it becomes a very big issue. Then let's go to solution for exercise 3. In this solution, we now say equation 1 is C1 V1 against C2 V2, where C1 is not given. C1 is not given, V1 is given, which is 2 liters. Mind you, this is total number of microbial cells in a watermelon juice, not the concentration. The concentration was not given. C1 was not given, V1 was given, 2 liters. That is the initial volume. C2 was not given in the question 2, while V2 was technically indirectly given, so that you use your logical sense to extract what is now V2. V2 is that if you read the question, they say 2 liter was the initial volume, and the final volume was you added 1 liter to dilute it. So if you have 2 liter plus 1 liter, total volume of liter for the final volume will be 3 liters, which is 2 liter plus the 1 liter of distilled water that was used to dilute it, which is 3 liter. Therefore, you will now go into the concentration. Note the number of cells. This is the number of cells. This is the number of cells. First of all, you must have to get the concentration of V1 for you to be able to continue with the equation. You cannot use 300 directly. So for you to get the C1, since the number of cells is given per volume, I told you concentration is number of cells per a particular volume. So you have to get the concentration of C1 initially before the 1 liter was added. For you now to get the C1, you take the total number of microbial cells, 30,000 cells, divided by 2 liter. So this 30,000 cells in 2 liter concentration in microbiology is measured in milliliter. Then you have to convert 2 liter. 2 liter will be equivalent to 2,000 ml. That is 1 liter is equals to 1,000 ml. Then when you now have 3,000 cells divided by 2,000 ml, then when you split it, you will now have 15 cells. 15 cells per ml. You have now determined the C1 in order to be able to use this equation. Otherwise, when you are looking for this C1 is not given, C2 is not given, you must have to determine the C1. So it's only C2 that you now use the equation 1 to determine. And in this case, you make the C2 the subject formula. Once you do that, you have C1 over V1 divided by V2. That will give you C1 is already determined here as 15, while the V1 is already known, 2 liter, divided by the V2. The V2 is, we already know how we got this one, into 3 liter. Then you now have 30 divided by 3. That will give you the C2 concentration is 10 cells per ml. That means once you take any cell, once you take 1 ml of this solution after dilution, you will have 10 cells. But once you take 1 ml before the dilution, you will have 15 cells in every ml. That is the calculation for exercise 3. Then this question says, note equations is for concentration of cell per volume, not the number of cell. The question gave you the number of cell, not the concentration, and you must use the concentration to do the calculation, not the number of cells. So if you have the number of cells, you have to convert it into concentration before you can use the equation 1 for this dilution process. Having said that, let's move to equation exercise 4. Let's take another example that will give us another insight on how to approach this dilution process in microbial analysis. It says, if a pot of soup labeled as containing 25 cells per ml of microbes in 50 ml of sample, what volume of distilled water is required to dilute the soup to a concentration of 5 cells per ml? This is a very tricky question, but very simple. If you be able to apply logic, you will get over it as simple as that. Moving to the solution, we say solution for exercise 4. Then we look into extracting the values. The values that were there was C1 is 25 ml, 25 cells per ml. V1 is 500 ml. C2 is 5 cells per ml. Why the V2 is not given? And the question is calculate. Look at the question again. The question is, what volume of distilled water is required to dilute the soup to a concentration of C2? We are looking for the volume of water that was added, not the total volume of water after it is being added. Then we quickly go into the C1. So the tricky question is that you must have to get the total volume of the C2, then you now from there you now know the volume that was added into the solution in order to reduce the concentration from 25 cells per mil into 5 cells per mil. That is the trickiness of the question. Please be very careful. Not that the question looks very simple, C1, C2, but there is, the purpose of dilution is for you to know how to play along with this thing and do the calculation very well. Having said that, first of all, you find the V2. Since the C1 is given, V1 is given, C2 is given, and V2 is not given, then you look for V2. Make V2 subject formula. When you make V2 subject formula, you have C1 the plus times V1 all over C2. When you extract those values and place them properly, you have 25 against 500 against 5. When you now do the arithmetic manipulation, you will now have 2,500 mil. That is, this V2 is 2,500 mil of total volume of water after that volume was added. Then the question now wants, you want to know how many volume was added to dilute this sample from 25 mil into 5 cell, or from 500 mil to this. Then you now say the volume that was added. Note, the question is what volume of distilled water required to be added. So the water that was added is the V2, the final one, minus the initial one. The final is 2,500 minus 500. So what was added into this substance in order to dilute the sample from 25 cell per mil to 5 cell per mil was 2,000 mil of water. This is the answer you are looking for, and not this answer. So if you stop here, your grading, your mark will not be complete. This is the final answer, so you have your complete mark. Having said that, then we'll look into other series of dilution in the physical class we'll be having. So I'll give you more of the questions so that we'll discuss that and have a better discussion on it. For the sake of time, I will want to end this class here so that we have a short note and discuss serial dilution, because that's a very big discussion on its own. I really don't want to waste much time. So though we have discussed a lot of it in the physical class, let's end the class. I will take up in the part C of lecture 4. That's why I told you lecture 4 or lesson 4 is the bone of contention, where we have to spend a lot of time and pay a lot of attention to really understand the basic calculation in microbial enumeration. Thank you.